Hello, everybody online. We're excited to have you, and we're excited to have all the people in person. So, my name is Mikayla Borton. I am a research professor at Colorado State University. And today I'm going to be talking to you mostly about DRAM and a little bit of review from yesterday and where we kind of are in the pipeline. And we're just going to go ahead and get started. Okay, so yesterday, Kelly talked about how, we go into an environment, we sample that environment, we get the DNA, and we sequence it. And then Ben took you through the process of going from reads all the way to bins, and we touched a little bit on dereplication. And so today, that's where we're going to start. So we've done metagenomic reads, we've gone through read trimming and assembly, then you have your assemblies, we then binned the contigs to get metagenome assembled genomes or MAGs. And now we're going to start with dereplication. And so the tool that we use for dereplication, and it is available in KBase, is drep. And so for people online and for participants here, that paper is in the participant materials if you want to read it. But I do want to give a quick rundown of how it works, and I'll do that in a second. And then following dereplication, then we have this set of MAGs that is our database, and we will annotate those MAGs with DRAM. Again, this paper is available also in the participant materials. And so really, once we have those annotations, then we can start to infer function from these microbial genomes. Okay, so the learning objectives for today. So I'm going to first talk about MAG dereplication. Then we're going to talk about annotation in general and the idea of annotation. Then we're going to talk about the DRAM workflow and how DRAM actually does genome annotation. And then we're gonna switch places, and Jared's gonna talk to you about how to actually infer function from genomes with your millions and millions of annotations that you get from microbial genomes. Okay, so if we start with derep or dereplication of genomes, here we're talking about the MAG level. So we have a bunch of MAGs. So each of you had a sample. We had 8 different samples. You all assembled and binned those samples. And so you can imagine that because they are all from the same system or same river, you might sample the same genome more than once. And so you now have this collection of genomes among all of you, and you may have sampled one genome. You may have sampled, so if we're talking about multiple samples, you may have assembled genome, the blue genome in both samples and the red genome in both samples, but you would have sampled them differently in each sample. So you might get a better representative from one sample than from another. And so the idea of dereplication is to assess which genomes are the same and then pick the one that is the best representative from that cluster. And so that's what derep does for us, is it groups genomes into like genomes and then decides which genome is the best representative for that cluster. And so how does derep run? The first thing that derep does is it runs checkm. So checkm, Ben talked about yesterday, which is a quality assessment of MAGs. And so it runs checkm to get all of that quality information. So things like completion and contamination. I do want to highlight here that the KBase default, so once it runs checkm, it filters your genomes and only keeps the ones that you tell it to keep. So the default parameters for those is it includes greater than 75% completion and less than 25% contamination. And so the whole set of genomes you give it, it filters based on these cutoffs, and then it goes through the pipeline of actually dereplicating these genomes. And it runs 2 clustering steps. So the first one is a MASH-based clustering. So it compares every genome to each other. And then the genomes that are 90% similar get into groups. And then it does a secondary clustering of those groups with ANI. And then it picks the best representative at 99%. I do want to just flag, so yesterday when we were talking about, I think there was a question in the chat of like, how do we decide what's a good genome? We're working with incomplete genomes. And so one of the ways that we decide that is through the checkm outputs. And so there is a paper by Bowers et al. And so these are the genome standards for MAGs. And I put this paper in the participant materials, but really what you have to pay attention to is table 1. So we do medium and high quality MAGs. And so a medium or a high quality MAG is greater than 90% completion and less than 5% contamination. It has some other stipulations, like it needs to have a 16S within the genome. And then medium quality is greater than 50% and less than 10% complete. And so with these defaults, and this isn't unique to KBase, it's the same defaults that DREP has in general if you were to run it on a command line, but just pay attention to these 75 and 25% are not the same standards that we're working with in MAG. So you might be inadvertently filtering out genomes before you even run checkm. So it's just something to pay attention to when you're working with all these defaults and running those tools. Okay. So then now we're going to go into assigning functions. So what we've done is you all had a sample, you all assembled, and then you binned it. You all have now a bunch of bins. You put them together into one place, and then you dereplicated them. And now you have a database of dereplicated genomes. And so we're going to now assign function to those genomes of dereplicated, genomes. Okay, so why do we need to know the function of genomes, of these microbial genomes? We really are interested in how microbial communities can impact the environment around them, right? And so we can do things like make these genome cartoons. So this one is one of Rhodobacter, and it is, using the TCA cycle and utilizing hydrogen sulfide. And so we can see that from a genome. And then if you think about microbial communities, there isn't just one genome, right? We already have in this room from 8 samples, we have, I think there's like 100 and some MAGs. So then that looks like this. So all of these functions then, we can start assigning functions to each genome. And so they all have a different function in the microbial community. So Geobacter is metal reduction, and we have obligate fermentation and sulfate reduction. And so we can start assigning functions to all of these microbial genomes using annotation. So what is the process then of annotation going from a genome to something like this genome cartoon where we know the microbial functions and how they could potentially impact the environment? So with genome annotation, we're first going to predict coding sequences. So we have those, remember we have those contigs, we have long pieces of DNA, and they're all grouped into MAGs. And so we want to first predict coding sequences. And so there's not gene predictions on these contigs yet. And so the first step of annotation, predict those coding sequences. And so we get these genes. From the genes, we then have sequences that we can compare to databases with genes of known function. And so these are databases like KEGG, UniProt, PFAM, DBCAN, all the different annotation databases, and we'll talk about those in a second. And then from these, we can just start to discern which pathways each microbial genome has. And that's really at the most broad level what genome annotation is. But what does this mean? So you have, a bunch of microbial genomes. Each of them have, on the order of more than 3,000 genes. And then if you hit each of those 3,000 genes to each database, then you have multiple annotations for each gene, and that starts to compile once you add tons and tons of organisms, right? And so understanding all of this data that gets input into annotation it's a pain. So how do we go from all of the genes, then these long lists of all the genes and all their annotations, and how do we get to function? And then I just also want to add, this is compounded by the fact that our databases are getting bigger and bigger and bigger and bigger. So that first paper I showed you, or the set of genome cartoons, that was I think there's, 50 or so mags in that paper. Now we're working on the order of 200,000 mags. And so this annotation problem and all the discerning function becomes a very large problem. And so okay. And then so what we've done then to go from that long list of annotations is we've built DRAM. So really there are a lot of annotators out there. There's, Proka, Ben went through a few of them yesterday. The thing that DRAM adds, in addition to just annotating all these genes, is it distills the functions. So it's not just a list of, genes and annotations. We go into specific process calling for each mag. And so we're going to talk about how DRAM does that today, and then we're going to use some of that data to do it. Okay, so conceptually, if you think about a annotation in a microbial genome, if the recipe is a genome and the genome or the genes are the ingredients, and we have something like chocolate chip cookies, this is the genome, and then the each ingredient is a gene. So we have 3 eggs, we have salt, we have vanilla, we have white sugar. We know exactly how much of each thing it takes to make a chocolate chip cookie. However, in genome annotation land, this is not always so simple. This is switching. Annotation isn't perfect, right? So there are non-specific annotations. So you might get like, oh, we don't know what kind of sugar and how much of each type of sugar. We might also not have information on, we just know it's white powder. We don't know what kind of white powder. Maybe it's flour, maybe it's not. There's also things that we just don't know. No idea. It's an unknown function. No idea. And so really, again, this is something we think about when we're talking about metagenomes, because this problem just becomes compounded and compounded. And so I'm hoping to show you today how we can overcome some of these barriers with DRAM. And also the idea that DRAM is a living annotation pipeline. And so for people who are working on specific pathways and you find genes that you are interested in, or you curate a gene set that maybe isn't in KEGG, we have ways in DRAM to adapt that so you can do your annotations at a higher level than maybe what the databases represent, because the databases are slow in catching up to all the science that we're all pushing out. Okay, so next we're going to talk about the specific workflow of DRAM. And so how does DRAM work? So this is the workflow. That paper is available. But the input is a FASTA of genomes or mags or assembly. So you don't have to be working at the mag level, but you do have to have an assembly base. So DRAM is not read based. It calls genes with prodigal. The gene calling step is done with prodigal, and then it annotates with a series of databases. And so it uses PFAM, KEGG, UniProt, CASI, MIROPS, and VogDB. It also does a lot of, it can input other types of data, which I'll talk about in a second. But it can also count for, in terms of quality, the number of tRNAs, and for that, it does tRNA scan. It can identify 5S, 16S, and 23S with BARNAP. And then also it does have the ability to input a user database, as I was talking about. So the data it ingests is things like GTDB and CheckIm, which Ben went through with you yesterday. So all of this is available in KBase, but you can ingest GTDB and CheckIm to just further understand how your annotations are related to the rest of your data set. Okay, I do want to just say that DRAM 2 is coming. So DRAM 1 is what is currently in KBase. DRAM 2 we're hoping to put out in the next few months. And there are a lot of adds to DRAM2. Specifically, we're adding new databases and new functionality. And the visualization part should be able to run on your own laptop computer, so it won't be server-based, and you won't need as much memory and things. So we're really excited about that, and stay tuned for that online. And we will be putting it into KBase as soon as we have it put out on GitHub. So I wanted to put this up here too. So Ben talked about yesterday different types of annotators in KBase. So Proca is one that he ran. And so these are just comparisons with DRAM. So these are all of the databases that DRAM uses, and this is a comparison. So DRAM does use more databases. Because it uses more databases, the runtime is a little higher relative to other annotators. However, the number of entries and the number of annotations is significantly higher also because we use more databases. However, because of that, our annotations are more complete. So we get more annotations per gene. We get more hypotheticals and less unannotated genes. Okay, so I've been telling you all, why DRAM is an annotator. It does do many of the same things as Proca, but where it is set apart from these other annotators is this part. So it does give you the raw annotation. So it gives you every annotation for every gene and every database that it hits to. So that's the raw. And then it also gives you these other two downstream products that are a derivative of this raw. So the distillate has distilled functions. So things like transporters, things like energy processes. And so all of that is housed in this distillate. So you don't have to sort through tons and tons and tons of annotations. And then it also gives you the product, which is an interactive heat map that Ben showed you yesterday in that KBase interface. And so really you can have a set of mags and not look at this raw annotation before deciding which genomes you're going to go after. So for example, if you're really interested in methanogens, you can see, from the product, these are all of our methanogens. I'm going to pull out these mags and I'm only going to look at these raw annotations. Or if I'm really interested in methanotropes, things like that. And so what does this mean? So if we take something like the human fecal microbiome mags, there's 76 mags. This is just a data set that we had on hand. This is about 40,000 gene annotations that you'd have to read through to understand all of these mags. That's a lot. Then if you go down a level into the distillate or distill, we get 881 carbon related genes, 650 transporter, 692 energy. And so then you can say, okay, well, in the human gut, I'm super interested in carbon, so I'm going to look at this part instead of having to wade through 40,000 or 140,000. And then if you want to go even further, you can go to the product, which is 98 metabolic products, which is in that heat map that Ben showed. I do want to stress that all of these data sets are linked. So I find something really cool in my product. I very much care about starch, let's say. I can then track that back through each level of annotation. And so I can go into the distillate and say, okay, this is green here. They called this true in the DRAM product because of these 4 genes in the distillate. Same thing in the annotations in the raw. You can then see, okay, I called this true here because of these genes and they're on this scaffold in my genome. I know I threw this in last night. I know there are viral people in the room. DRAM-V is for viruses, and specifically it has much of the same annotation pipeline, but what it does also have is prediction of auxiliary metabolic genes. And so that is also running in KBase. But I just wanted to flag, I'm happy to talk about it more. This is more focused on mags, but if you're interested in viruses, I can talk to you about DRAM-V. And then one of the other things I wanted to flag, the only difference between DRAM, running DRAM and downloading DRAM, from something like Bioconda is, and KBase is that DRAM in KBase, uses KOFAM. So instead of KEG database, and the reason for that is it's a licensing thing. However, the data here is, comparable, most of the data, and actually you can run KOFAM, it runs much faster outside of or in DRAM also, compared to running it with KEG. So, but just something to be aware of. If you're not using KEG proper, if you're running it in KBase, you're using KOFAM, and it's just something to consider for methods and things. The other thing I wanted to highlight really is that the benefit of DRAM being in KBase is that it's linked to all these other tools. So having all of these annotations within KBase, you can put your genomes in, you can run the DRAM annotations, and then you can put them into something like genome scale metabolic models, because there are so many tools on KBase. And so being able to use DRAM annotations in something like a genome scale metabolic model, KBase is the only place that you can really do that, outside of being able to code it yourself. And then the last thing I just wanted to, so DRAM is like a labor of love from a lot of people. So it's not just me, it's not just Kelly, it's our whole lab. And so we have lots of people in our lab who have put a lot of work into this. We have a lot of people that we've worked with at Argonne, Denver, University of Queensland, Ohio State, and then many of the other national labs. So I just want to highlight here that this is a work from everybody, not from just, my lab or Kelly's lab. And then I'm gonna hand it off to Jared. Question. Do we have time for questions?
Yeah, I think we have time.
Cool.
Okay. They should be able to hear me online, so if you can't, just shout it out. But we have a question. Can we also get gene expression level through DRAM?
Not currently, but it's in DRAM 2, which we're really excited about. But yes, the DRAM does output different files, GFFs and things that you can go ahead and input into something like a expression mapper. But it doesn't do it on its own. It is coming though. Other questions? There is a few minutes from, yeah.
I was going to get like COGS in the DRAM too.
COGS are not in DRAM 2, but I would like to write that down. I will, take that.
Be like 15 unique organisms to really figure out what different things could be shown to do in DRAM and what unique physiologies could be represented by DRAM. So we're going to talk about mock community that includes things like a metanogen, like Metanosarsina, and some nitrate reducers and desulfobacterium. And we're just kind of walk through what their DRAM annotation is going to look like, so you'll see what the products look like. The next, please. Thank you. So Kayla just walked through this, so you know that when you're running DRAM, you're going to annotate your genomes with DRAM. It's going to use those 6 different databases Kayla talked about to annotate your mags.
Oh, sorry.
Thank you. And like Kayla mentioned, you're going to get 3 really pieces of information or products out of DRAM. You're going to get the raw, the distillate, and the product. We're really going to work backwards and focus on the product today, because the product is really the most quick to read, user-friendly, quickly informative output of DRAM. But really the raw you're going to get is just going to be your actual annotation files. So you're going to get a sequence file of nucleotide data. You're going to get one of amino acid data for all your genes and protein sequences that are called. So there are definitely times where you want that actual sequence data, and you'll have that output by DRAM anytime you annotate your mags. The distillate is really what underlies the heat map of the product Kayla kind of just showed you. For the distillate, you're going to get a couple files. Probably the key one is called the metabolism summary. And you're really going to get a very well organized Excel spreadsheet where you have like 6 tabs on there. You have tabs for carbon, for energy, for transporters, and you're going to see a huge number of different genes. And then the metabolism summary in there is going to tell you basically for every gene in that spreadsheet, how many copies or homologs does your individual genome encode. And this is not a run one at a time thing. You can run thousands of mags through your genomes through DRAM at a time. And the product is the heat map that we're gonna really walk through today. It's that really user-friendly, quick thing to scan. So we're gonna kind of go step by step of how to work through the product. Please, the next. Thank you, cool. So this is what the product looks like. Orienting you here, every row on this heat map is going to be a different genome. So in this case, we have our 15 different ones. We're probably going to focus on this blue Dichloromonas or aromatica as our kind of model one that's just highlighted on the slides here. But every row on this heat map is going to be a different genome or mag that you've run through DRAM. And then every column is going to be really the gene content you're looking for. We're going to go piece by piece here, but in some cases, you're going to be talking about pathways like carbon fixation are the columns here. In some cases, we're talking about individual protein complexes and a bunch of subunits. And then in some cases, those columns are going to represent individual genes or proteins. So we'll go step by step through these. Perfect, thank you. So first thing DRAM is going to tell you is pathway completion for carbon fixation. So, I guess one question I will throw out to the group if anyone has any ideas is if you're trying to figure out maybe how your genome or your bacteria makes energy, if you're looking for the presence of a pathway, like, would you say that you need to have a complete pathway present or 100% of the genes in a pathway to call something as having potential for that? Does anyone have any thoughts on that?
And remember, you're dealing with, not always complete genomes.
Yeah, so you have environmental genomes. Do you think you always have to find like a complete pathway to something to have potential? Cool. So I see most people nodding no, which is perfect. So really think about the fact that you have to be a little bit flexible, that if you have an environmental genome and it's your mag only like 78% complete, you're probably not going to have the complete genes for every pathway of interest that you're looking for. But you can still say your mag has potential for something like glycolysis. So one thing DRAM is going to do is that really for the carbon fixation pathways. So if you can't read this, we're talking glycolysis, the pentose phosphate, TCA cycle. It's going to give you pathway completion. This is a heat map where basically the darker you get, the greater percent completion your mag encodes of that pathway. There are also cases for some of these pathways. Over here, you see glycolysis, where you might have variations on a pathway where you can have glycolysis in 9 or 10 steps, depending on what your organism encodes. DRAM is going to basically look at all variations of those pathways. It's going to figure out what percentage of every variation your genome encodes, and then whichever is higher, it's going to report back in this heat map. Is that okay with everyone? Perfect. So the first thing, DRAM is going to tell you what percent of that pathway your genome encodes. It's also
Okay.
It's also going to tell you then for really the components of the electron transport chain, so complexes 1, 2, 3, 4. In this case, instead of pathway completion, you're getting the really subunit completion of these multi-subunit proteins. The idea here, and we'll talk about respiration in a minute, but if you're a respiratory organism, you need to encode an electron transport chain. The proteins involved in that, at least in the case of like the NADH dehydrogenase, encode like 14 subunits. This is a case where instead of thinking that your mag might not encode 100% of the pathway, you might not encode every component of that protein, but if you have most of the subunits, if you probably have a majority of those subunits, you can say that your mag encodes complex 1 or NADH dehydrogenase. So there are a lot of different variations on complex 1, 2, 3, 4 in the electron transport chain, and DRAM is just going to tell you what percent of those multi-subunit proteins your mag encodes there. Where this matters, one example highlighted here is there's a case where if you encode NADH dehydrogenase, again, that is 14 subunits. If your mag encodes like 50+% of that, it probably encodes that real protein. But there are cases where things have a trimeric hydrogenase that is homologous to just 3 of the 14 subunits. And if you see a pattern where your genome just has 3 of those, it's only 21% of that, it's probably something else. So you need to be maybe flexible a little bit with that 50% cutoff, but you have to be careful not to probably overinterpret it. Do you agree with that? So in addition to those percent completion for pathway and multi-subunit protein heat maps, DRAM also gives you this kind of binary presence absent heat map of really marker genes. In this case, they're marker genes for other really electronic sectors, so things like nitrogen metabolism, sulfur metabolism, photosynthesis, and methane. So in this case, if something is highlighted as green, your genome has it. So in this case, your Dichloromonas is going to have this gene here, which is for nitrate to nitrite reduction. If it's green, it has it. If it's missing, it's absent in that genome. Go next. Does anyone have any questions at this point on the heat map? Yes.
So do you always start with the percentage completeness and then you move over to your presence and absence?
I would say based on what your question is.
Repeat the question.
Okay,
Oh,
the question was, do you kind of always start by looking just like the pathway completion part of the heat map and then go over your presence absence? And I would say it's very much tailored to whatever you're looking at, particularly if you're interested in carbon metabolism, if you want to look at just nitrogen. Some things like Kayla's kind of alluding to are not represented in this. We're going to put it in DRAM too, but it's really, yeah, targeted to what you care about. Yes.
Can we also assume that if a pathway is absent, could it mean that it's not just sequence, it's part of the missing molecule?
Yeah, absolutely. So this is a case where if you see, so the question is, if you see that a pathway is absent, can you assume that it's kind of just part of a missing sequence? This is a case where, yeah, if you think your organism should possess like glycolysis, but you see it's absent, but you go look at the completeness and your genome is like 50% complete, yeah, that's a case to be careful of. So exactly. Yeah.
Presented by relative abundance of the gene. So is this based on that or like if you want to
Will you repeat that louder?
also usually you see these outputs are presented by relative abundance of the pathways with absence of presence.
Yeah.
You can now I'm asking if the presence or absence is represented by a similar heat of the presence.
Yeah, so the presence, so the question is really, is there anything about relative abundance underlying the presence absence, or is it just based on one copy? Yeah, so this is really just saying there's at least one copy. If you go to the metabolism summary though, you'll see in that case, if your genome has just one copy or if it has 4, it'll tell you there. So that's where they go into distillate and see exactly how many copies you have. You can go deeper into it. But yeah, this is just saying at least one. Yeah.
questions online? You good?
All right, so one really nice feature of this product too is it's an interactive HTML file. So if you are looking at your heat maps and you see that your dechlor modus encodes one of the nitrogen fixation genes and you want to know exactly what that is, because here it just says nitrate, it doesn't tell you more specifically, but you can hover over that, you're going to get a little box popping up. And that box is going to tell you what genome this is from. Your genome name is going to tell you the category of metabolism, in this case nitrogen. Going to tell you subcategory of denitrification, so to be organized into kind of a specific pathway for that metabolism that is in DRAM. And it's also going to give you the function ID, so it's going to give you the KO if there is one. So then you can go to KEG, you can look that up more specifically and see a lot more about what that protein does, what that gene does. And then it's just going to give you like the EC number, some other information, but you can hover over and see a lot more about exactly what that gene is in your MAG or genome. So one question you can have then when you're looking at a genome is what types of energy generation metabolism are encoded in your genomes. And that's one thing you can fairly start with the product. Not every form of energy metabolism is in the product, but we're building more in. You can at least take a really good, I'd say, stab at the most common environmental metabolisms probably here. Perfect. So when you are trying to read through a genome for energy metabolism, probably one of the first questions to ask is whether your organism is respiratory or fermentative. If you're looking for things that do respiration or respire, the key hallmark to look for is if your genome encodes an electron transport chain and a Krebs or TCA cycle. Things that respire, they're going to have that electron transport chain, and they're also going to have genes to use some kind of external electron acceptor. That might just be the genes or proteins involved in like oxidative electron transfer, but you might also have genes for the use of nitrate or iron or sulfur or things like that. And organisms that do this, they're going to make their ATP via oxidative phosphorylation. So they're going to encode the proteins that are needed to kind of make a proton motive force. They're going to have some kind of ATPase3 that's involved in that like, classic electron transport chain. We'll hit next. However, if you have an organism that is a fermenter, you're going to probably diagnose that via an absence of an electron transport chain in the Krebs cycle. One thing I would caution you probably here is that because you're diagnosing things based on the absence of that electron transport chain, this is probably just a case where you want to look at genome completeness again. Because if you're looking for an absence, but again, it turns out to be a really lowly complete MAG, it's only 20% complete, you can't really say much from that. So I would just be careful and look at that metric in conjunction with the gene content from DRAM. Right, so if we're going to kind of model walking through a genome to see whether it is a respire fermenter, you see in this box, pretty much the red boxes are highlighting your respirers here. But if you look at this Dichloromonas, this is the TCA cycle highlighted in orange, and you see that it's very dark blue. It actually has about 100% complete TCA cycle. But if you scroll through everything that's highlighted as a respirer, in this case, looks like they all have the complete TCA cycle. But even if it looked like it was closer to like 80% of that pathway, I think it would still be fair to say it encodes that. But then if you look over at your complex 1, complex 2, complex 3, your members of the electron transport chain, you're going to see this dark blue. Everything in this red box has pretty much 100% completion of the subunits of that NADH dehydrogenase. So these are respirers. I'm going to kind of point out this methanosarsida acetoborans. If you look at it, does encode the TCA cycle, but it doesn't really have anything involved in the electron transport chain. So that's kind of where you can start to see in that heat map where things are missing that. So that's, we maybe don't know what that is from this yet, but we can say it's not doing respiratory metabolism. Is that okay with everyone? Cool. Move on. After you look for respiration versus fermentation, you can try to think about what energy source something's using. Is it chemical? Is it light? And you can also diagnose those with DRAM because that presence absence heat map is going to tell you if things have the gene for photosynthesis. Are they using light? Or are there other genes for these other chemical electron sources? Yeah. So you can look over here, and if we have our Dichloromonas that we said was a respirer, then we can go over here and look at the nitrogen genes, and we see it does have some of the genes for use of nitrogen. In this case, it also can use sulfur, but it has nothing for photo. It doesn't have any photo systems, so we know that can probably reduce nitrogen and sulfur, but it's not photosynthetic. Is that okay with everyone? Perfect. So you can really go case by case with that. DRAM also includes other metabolisms. So we have methanosarsida acetoborans on here. It's a methanogen. So we said that looking through, we saw that was not a respirer. It does not have NADH dehydrogenase. It doesn't really have genes for sulfur or nitrogen use, but over here we have a methane category, and it has basically every category, or it is gene for every form of methanogenesis. One really nice thing of the methane category also is that the first thing here, we have the functional gene, we have the marker gene of methanogenesis, which is just MCRA. So if a genome has that, you might call it a methanogen, but DRAM is also going to subdivide methane gene content into methanogenic pathways. And does it use acetate or trimethylamine, or does it use carbon dioxide and hydrogen? So DRAM will kind of give you deeper information on methanogens too. Another thing DRAM will give you, and Mikayla brought up enzymes a little bit, but it will also start telling you what carbon sources things can use. Everything needs a source of energy, but things also need a source of carbon. DRAM has a category on that binary heat map for enzymes and one for short chain fatty acids and alcohol conversions. So here you're just going to see, do your genomes have the genes encoded for alcohol production? Does it use butyrate? Can it use lactate? There are a lot of just really common short chain fatty acids that are probably pretty frequently important to people's work. You can see that if your organism encodes there. And then for the enzymes, these are carbohydrate active enzymes. These are really what are used to break down like large polymeric carbon, things like starch, things like mucin or pectin are on here. And again, this will just give you the presence absence gene content if your genome encodes those. Cool. So DRAM does have a lot of information on these carbohydrate active enzymes or enzymes. So these are really, again, enzymes to break down. They break glycosidic bonds, which are just sugar-sugar monomeric bonds. These are really for large polymer degradation. We have a lot of these in DRAM. This is a really expert curated feature that we have in DRAM. DRAM basically uses DDCAN2, which is a very expert curated database for enzymes or these large polymer carbohydrate substrates specifically. This would have next been curated by Phil Pope and his former postdoc Sabina. So they took a lot of time, used their expertise to really guide our group on how to build enzymes into DRAM. One really nice feature here is that for things to be called really users of these large polymeric carbon, they need to basically be able to take a polymer, cleave a shorter piece or shorter oligomer off of that large backbone of the larger polymer, and they need to be able to then degrade that oligomer into like a small monomeric piece of carbon or sugar. Is everyone okay with that? So the DRAM diagnostic, I believe, to be able to utilize these substrates is that a genome must encode enzymes both for the backbone cleavage step and the oligo cleavage step. So it can do both to be able to completely take that large polymer, break it down to its smallest part, and then use the smallest part. Next. Are there any questions before we kind of summarize all the enzymes or anything?
Really, that is, I think the Kzymes are the perfect example of how DRAM is different from like Proca, for example. That was a lot of manual curation, and also we required the oligo and the backbone because you can make a lot of those Kzymes are promiscuous, so they act on multiple different substrates. And so really these are really I guess really lowering our false positives for calling these genes on larger carbon. And so we spent a lot of time there, and that is an add of DRAM that other annotation tools don't have.
So then summarizing really what you get out of DRAM and how you can understand your organism's metabolism from the data you get out of here. From the product, the heat map we walked through, you can really start to build a pretty clear picture of what your organism has the potential to do. You can understand what its energy metabolism might be and what its carbon metabolism might be. But we know that there's going to be a lot more information in a genome than just those two things. So that's about the complexity you get from the product. But if you look at the distillate, again, is that Excel spreadsheet that kind of underlies the product. There is a lot more in the distillate than is represented in the product. The distillate will tell you like what actual subunits of the electron transport chain your genome encodes, and it will tell you which genes of glycolysis your organism encodes, but it will also tell you different transporters, antibiotic resistance genes. It has CRISPR-Cas in there. It has a lot more underlying that. So if you look at the metabolism summary in the distillate, you can start to build a deeper picture of what your organism has potential to do. And if there are like particular features of metabolism or the lifestyle strategy of your organism that you're interested in that aren't in the product, there's a very good chance they're in the metabolism summary. So you can start to build other things like metal transport onto here, or whether your cell has a flagella or not. So you can kind of take your analysis and go a step further. But then not everything in the genome is represented in DRAM yet, but every gene is going to be annotated by DRAM. So if there's something you're interested in looking at that is not even in the distillate, it really should be in the raw. It should be in the annotated genes. Then you still have that annotated gene file that you can then go manually look through, look for whatever your metabolism or genes of interest are. And then you can still, in this case, look for something like a multi-heme cytochrome that might be something novel, might just be something that's not in DRAM, but you still have the ability to go into the data file and figure out what your genome has potential for. And then just to show you what the output of DRAM is going to look like in KBase, the product.html is going to be that heat map file, that interactive file that I would probably suggest you start looking at first. It's easiest to look through, but you are going to get a lot of different files. You're going to get an annotations file. You're going to get your actual genes, nucleotide amino acid sequence files, and that GFF file Kayla talked about. You're also going to get files with the rRNA and tRNA sequences, the number of rRNA and tRNAs that were identified in your genomes. Your metabolism summary is going to be that kind of distillate spreadsheet that underlies the product heat map and tells you really extensively what is in your genome. And you'll get all of these every time you run DRAM. Any questions on this? Because that should be the last slide here. Yeah.
The first one is, when you go back to the slide.
Oops.
And I wanted to know, there's this nice diagram that you showed us with all the time. Can we get that out from this one?
Like this?
No,
Or the cartoon?
Can we get that out? How do we get that?
Oh, how do you
Yeah,
get this?
that's a
Yeah.
manual.
Yeah, so
Yeah.
this is a function that we often get asked for, but yeah, this was made from all of the outputs of DRAM. We made this in Illustrator. But that is something that is probably a year in the works. We really do want to have something like a genome cartoon feature of DRAM.
I have a follow-up question.
Sure.
If you look at the heat map, sometimes like what I noticed with like Max, there's, I think they call it microbial cooperative specialization. Is it possible to also draw a conclusion that with a certain pathways, we have a group of mags that are carrying this task and another group is carrying another task, and overall the pathways are happening because of the various tasks? Can we draw that conclusion with this type of data?
And I think especially if you, yes,
Okay.
so really, are you able to see kind of microbial cooperation from that heat map if you have different members of your community that do different things? But say if you run, especially if you run like tens or hundreds of thousands of genomes through DRAM at once, you're going to see patterns follow on that heat map pretty well. So I would say, yeah, you should be able to see that kind of cooperation.
And also just to say, so this, so that product heat map is an HTML. It's not currently sortable. That is a feature that's coming out in DRAM 2. We're going to have like a sortable heat map. So you could be like, I want to sort by methanogens or whatever. But also in the DRAM output, that product is available in a TSV. So you can easily put it into something like Excel and like sort that thing by like, a case that is a good example in the product heat map of what you're saying is the nitrogen cycle. So you could sort by each nitrogen cycle and see which organisms are carrying out each step. Okay. Is it on the chat? Oh. Okay.
Yeah.
We have a question on Zoom.
Yes. Can you hear me?
Yes.
Okay. Yeah, so I have a question when you have which is a bit related to monitoring. So say if you have these mags assembled from multiple samples, can you map back these functions to the original samples? Do you have a view for that?
We don't have a view for that currently. It is something that we're working on. I would say a way right now, if you wanted to do that now, what you could do is map to the genes.f. I don't know what type of data you're talking about, but if you have reads, you could map to the genes.fna. Or if you have something like metaproteomes, you can map to the genes.faa. And those files all have the same headers that then can be linked back to the annotations file or any of these other files. But the point is that all the files are linked. So if you map to something like the genes, using those headers, you'll be able to easily link into the annotations and see per sample abundance profiles.
Okay. I have to think about it. Yeah.
Yeah, but it is something we're working on, because we know personally, even in our lab, it is a thing that we routinely do, and so we want to automate all the things that we routinely do, and so this is something we're definitely working on.
Yeah. I mean, it's, for example, if you do soil analysis, you want to see if methanotrophy is present in one soil type versus the other. So that kind of stuff.
Yep.
But maybe you have incomplete metagenome from one sample, which is completed by another sample. So yeah.
Totally. Thanks for your question.
Okay, thank you for your answer.
Go ahead.
Yeah, so what DRAM 2 coming out, what are the additional functional categories that you're adding that make you most excited about, and also where should the gaps still lay as far as libraries and things like that
So in terms of the functions, I'd say that I am most excited about for DRAM 2 are having the ability to overlay abundance onto these heat maps. That is like something we've needed for a while, and it is something that every time I teach someone DRAM in our lab, they're like, what about this? I'm like, yeah, okay, this is the script. I have the script. This is how you do it. But like, we need to make it automated in DRAM. So that is something I'm really excited about. We're also really excited about the virtual or the HTML, the products, because they're more interactive. So you'll be able to be like sorting by a function, or you might be able to sort by a taxonomy that you care about, and really output different versions of the heat map. It won't just be the static thing that gets output. And then in terms of functions that I think we are both really excited about is Jared has built a, do you want to talk about your methyl for,
Basis, so I focus on like methylotrophy or metabolism of methylated compounds. So we're really, especially also for the sake of trying to better annotate like methanogenic physiological diversity, we're trying to include like a methyl annotator. It's kind of one of the metabolisms we've been mixing in
yeah.
that space.
And they're not in databases. And so another one that's really exciting is Bridget's, which is on, she built a module on polyphenolics. Again, those are things that like she built the HMMs for that aren't in databases. And so that's something that's really exciting. And then the last piece that we're working towards in DRAM and is coming is that ecosystem specific products. So in the human gut, you might not necessarily care about, you know, iron reduction, but you do really care about sugar transport and things. And so we're setting up the products so they're ecosystem specific. So we'll have one for soil and we'll have one for the gut and we'll have one for engineered systems because, you know, people really care about, you know, different types of hydrocarbon degradation. And so those are places that we're going. And also I just want to flag for people online and here, if there is a system that you really care about, we're always like, we work in, you know, soil and rivers, but if you're an expert in a system and you're like, man, I really would just like to have this output, we're totally willing to work with people to build the outputs that they want. So as long as we can just like make it something that is usable in DRAM for everyone. And so that is a thing I just want to put out there. If that's something you're interested in, we would be totally willing to work with you on that. Yeah.
Any chance that it's going to be doctorized?
Yes, it should be. Yes. We had a question in the room, if it would be Dockerized, and it will be.
Okay, we good? Awesome. So yesterday, right before the break, you had taken a quiz about what microbe you were. So I wanted to share our results. So we're coming from so many different places across the world, both in person and virtually. And you can see that as well in the total diversity of our microbial communities. And I also wanted to do a little comparison between our two sample sites, since we do have in person and virtual. So we have the total diversity here. And then you can see that our communities are very different between our two sample sites. So there are definitely some factors here which are bringing out some different, I guess, like relative abundances of our of our different microbes. But you can see that the Elphidium is quite a large representation in both. But then you can also see that virtually we have a few microbes that aren't represented within the in-person community.
Interesting concept around sample bias.
Yeah. So just wanted to share that before the break, and I will. Nothing for online folks during break. I think we'll start at.
Okay, so 20-minute break.
Okay, 20-minute break. We'll see you all in 20 minutes.
Okay, we're gonna go ahead and get started next with a presentation from the National Microbiome Data Collaborative.
Yeah, good morning, guys. I'm Ellenbogen. Microbiome Data Collaborative. And today we're going to talk to you guys about. Can you talk a bit louder, please? Okay, thank you. So, and today we're going to talk to you guys about the NMDC and then the sample metadata, which is part of the ISME 2024 section. So, next slide, please. So my name is Ian Oduwole. I'm a PhD candidate in the Genomic Science and Technology program from the University of Tennessee, Breslin Center. And my research interest lies in bioinformatics and metadata analysis. I particularly focused on applying machine learning and other bioinformatics techniques to study uncultured microbes, and particularly focused on the extreme uncultured microbes, particularly from the permafrost environment. So we're trying to see
how we can culture more microbes in the laboratory by studying their genomics as well. So, and here is Emily.
I'm
as well.
Emily. Nice to meet you all. I'm a postdoctoral researcher at the Scripps Institution of Oceanography in San Diego, and I do a lot of bioinformatics. I was once in the position of banging my head against a table, figuring out how to do metagenomics and all these bioinformatic things. So I think KBase is so amazing. It's what got me involved in the NMDC program to try to communicate, ways where we can make this data and everything more accessible and understandable. I work on understanding microbes and viruses in extreme environments. So that includes super salty lakes, some crazy microbial mat communities, and even in some watering pans up in Botswana. And I have, a lot of questions and curiosities about evolution of life on Earth, and I mainly use bioinformatics and metagenomics to study and answer these questions. And I'm just going to take the rest away, and I will be back later.
Thank you. So here is also Ken. Ken is a PhD candidate in the Department of Plant and Soil Science. He's going to be on the video trying to talk to you guys about the sample metadata. So I will probably need him to introduce himself as well. So yeah. So today I will just introduce you guys to NMDC, what we do, our engagement activities, our sample metadata, and like what everything is all about. So basically, it's more of engagement activities. We are not very different from KBase, but the purpose of NMDC, you can see, is data collaboratives. We are trying to build a research community where researchers can actually come together to talk about data, exchange data, reuse data, make the data accessible to all other research communities as well. So we are going to talk to you about what we do, as I said, how you can standardize your data, the importance of doing that, the activities that we have for you guys, and how you guys can actually also get involved. So the purpose of this training or this workshop is that we want to give you the benefits. We want to provide an overview of metadata standardization and how you can actually promote interoperability. It means that how you can actually use data to communicate to other research community, and as well as make the data access to all other people in other research areas as well. So we're also going to also discuss the importance of metadata standards, which would enable data reuse, because we believe that if the data is standard, then everyone can have access to data. If everyone is using the same template, then everyone can understand the data, because we all go to the field, we generate data, and then we write it in different formats. And then, sometimes when scientists use like result from other, maybe other research community, it takes some time to understand. So the purpose of NMDC is to give everyone the same template, the same splice for them to understand the data. So we also introduce you guys to the NMDC submission product as well. So as I said, what is the NMDC? NMDC stands for the National Microbiome Data Collaborative. It's a microbiome data sharing network that actually supports data standards. It gives robust infrastructure on how you can standardize the bioinformatics workflow of your data and for community building. So it focuses on three main aims. The first one is the submission product. So where you can contribute and share your own study and biosample information. So one thing that NMDC is unique about is how you collect your data, how you submit your data. So they want a procedure where if you submit your data, someone doesn't have to contact you to use your data, and where you can communicate the same language of data with other researchers without having to, go through your files again. So see, okay, how did I store this? How did I store this data? What format did I store this data? So they have this standardized format where if you process your data to maybe JGI or other facilities, you can actually have access to that template, and you can store your data in that format. So we also have the NMDC Edge, which helps to process your data with its standardized bioinformatics workflows. Basically, everything you can do with NMDC Edge, you can also use KBase. But one thing about the NMDC Edge is that NMDC Edge gives you a standardized bioinformatics workflows, which means that it gives you a workflow where you can just run your data without having to select to do multiple clicks. So it has a, for example, if you are running Max workflow, So all the software or the tools that are involved are standardized. So you don't have to select anything. So in that way, it means that if you use a particular tool and other scientists that are working in the same field also use the same tool, you're probably going to get the same results and communicate in the same language. So for example, in KBase, it's possible that you have like several tools that you can use for your mag annotations and stuff like that, you can select. But for NMDC Edge, you just have to use one tool. One is only available for you to use, which means that the tool that you are using is the same or similar tools that other scientists are using. And in that way, you can get similar results if you work in similar data, with similar data sets. So, and the last one is the data portal. So the data portal allows you to actually discover and access standardized multi-omics microbiome data. So what NMDC does is to kind of compile this data in different sets. For example, if you're looking for data in a particular environment, so you can have access to all the data involved, like the metagenomics data, the metaproteomics data, the soil organic matter data, and all the data that are involved in that workflow. So basically, any of the workflows that you run on NMDC, any of the data is actually findable and accessible to any other scientist without any hassles. So NMDC, as I said, is committed to the principle of what we call FAIR, which makes data to be findable, accessible, interpretable, and reusable. And this is exactly one of the problems with people working with data. Sometimes you read papers, and then for you to access the data in that paper is very difficult. Sometimes it may be stored in different database where you don't have access to. And in that way, it actually limits your your communication with our research, your experience of using other people's data. And in that way, we may not be able to kind of find new studies or be able to compare our data or our studies with another scientist. So what NMDC prioritizes is that it prioritizes standardization and user-centered design to achieve this fair and open data. So the first one, as I said, is it makes data findable, accessible, interpretable, and reusable. So these are all the four key or all the four concepts that NMDC stands for. So in this way, NMDC is able to support microbiome discovery. And one of the discovery of microbiome is that NMDC has a lot of collaborations with other facilities. For example, with the World facility, the NION, the NSF, and then the microbiome project. So the data set from all these facilities are also available for NMDCs, but in a different format, in a way that it's been standardized, where you can actually access the data and then compare the data set to other studies without any struggles. So one way is that NMDCs can support, for example, the question of what role do microbes play in the percent of soil carbon? For example, if you're a researcher that is interested in this kind of study, you can actually collaborate with other researchers in similar area. You can have access to their data, and you can be on the same page when asking these kind of questions. Another question is, how do microbes mediate watershed scale nutrient transformation? This is an example of one of the focus that NMDC is interested in. If you are a part of researcher that is also interested in asking some of these similar questions, so you can access similar data sets from NMDC, and you can maybe you're interested in getting some mags, you're interested in getting some metaproteomic data, so you can have the access to this data set. You can have the access to the scientists that are involved. You can collaborate with their data. You can use their data freely without having to get permissions and stuff like that. So another one is, if you are interested in how do microbes impact biogeochemical cycling locally and across the continental scales. So this is also what NMDC does. You can talk to other scientists working in this area. You can use the data sets in the same format without having to format to do any kind of reformatting or reanalysis. You can access the data in the whole format, and you can also contribute to the data. So as I said, what NMDC does is to support new ways for researchers to ask questions, compare across studies, and reuse data from other scientists. And in that way, if you have access to data from other scientists, it actually generates novel questions. It generates novel ideas. Because, for example, if you are seeing a consistent trend across maybe an environment, maybe you are working in maybe hot spring environment, and you're seeing a consistent trend in your data set, and in the data set of other scientists, it actually brings more compelling conclusion from that environment. And that is what NMDC has to offer. I remember just from this, I remember when I was working with one of the data sets that has like, I think it's from probably 10,000 environments, and we have to write like emails, getting permissions from each of the scientists having the data sets. Just okay, hey guys, can we use your data? Can we use your data? Can we use your data? And you know, it's created a lot of hassles because writing 10,000 emails, we have to kind of use a Python code. Okay, just send these general emails to everyone and see like people that give us permission. And in doing so, it's actually limit the time and you know how we can actually operate with that data. And with that barriers, if you're using NMDC, you're not going to face that barriers because it's stipulated that your data must be accessible, must be fair, and must be open, must be an open access data to everyone. So so the 3 product initiatives of NMDC, if you are interested in like exploring their website, the first one is Summation Portal, which actually lowers the barrier to collect and study and biosample data. So what NMDC is actually developing right now is to give you a format that when you go on the field to collect your data, maybe you're going on field trips, you'll be able to collect all the essential parameters for that data set. So that when other scientists see those parameters, they will be able to use those parameters in the data set. For example, maybe they are going into the ocean to collect some, you know, some samples. So NMDC will give you the templates. I think they have an app now where you can just download the software. They'll give you the templates of what samples, what parameters that you need to collect, with the temperature, the pressure, the pH, the salinity in the white formats. So that when other scientists see that format, they don't have to say, oh, how did you store your salinity? How did you store your temperature? Is it in degrees Celsius? Is it in Fahrenheit? So you guys can easily, you know, have that easy communication, even if you are not in the same research group. So the second one is the NMDC Edge. So the NMDC Edge is streamlined multi-omics data processing. So it's very easy to use. As I said before, anything that you can do on NMDC Edge, you can do it with KBase as well. And what NMDC Edge offers is that it doesn't give you varieties of tools. It only has a set of tools where you can just submit your data set and then it runs through the standardized bioinformatics workflow. So the workflows are regularly updated. So I'll probably see that a benefit for people that don't really want to like have a lot of hassles or stress in trying to say, okay, which is the best tools for this, which is the best tools for it. So NMDC hover like the kind of overall package like a pizza, where you know it has all the tools inside, and then you don't have to select any of the tools, just use the best tools for this, and then you get your results once. So the data portal and API is that it gives you the access and discovery of microbiome data information. So as I said before, NMDC, you can also have access to a lot of data sets in a standardized format. And when you have those data sets, you can get a lot of information from them. You don't have to you know, go from one side to get this data set and go from another side to get another data set. So from this information, you can get what data set that are involved and, you know, you can have access to them as well. So how you can engage with NMDC? Emily and I and other ambassadors are part of the ambassadors for this year NMDCs. So they regularly run the ambassadorship program where you can have, if you're interested, if you're passionate about promoting data stewardship and data management, if you are passionate about metadata standards and multi-omics data processing, so you can be part of the NMDC ambassadors, where you don't necessarily have to, as long as you're working with microbiome data, then you could be part of the ambassador as well. So, so we're going to talk about what are metadata, and this is going to be backend. Thank you.
Okay, thanks. All right, I'm going to switch over to the video.
Good afternoon, everybody. Hope you've had a good KBase workshop so far, and sorry I couldn't make it there with you all, but hopefully everything is going along well and you guys are still have enough energy to hear my section about metadata and metadata standards. So my name is Kent and I'm an NMDC ambassador, and today I'll be giving you a brief rundown on what metadata is, metadata standards, and hopefully explain to you why it's so important that you are able to capture good metadata for your project, as well as for anybody who might want to access your data down the line. So before we get into the meat and potatoes of metadata, I'll give you a little information about myself and why I think metadata is so important. So right now I am a PhD candidate at University of Kentucky, and my research focuses on the intersection of agriculture and microbiome sciences, particularly metagenomics. So my research is two-part. One side of it is the standard omics pathway, where I'm going out into the field, collecting things like soil or plant roots, plant stems, taking that back to lab, sequencing the microbes held within my sample, and then trying to analyze the community. And then the other part of my research is actually analyzing the field, field conditions, and other environmental data that might come back and shape that microbial community. Now, since all of these factors are interrelated, it's essential that I don't skip any steps, and that any data that I'm collecting, whether it's my primary omics data or the metadata, that all of it is robust and well documented, and most importantly, standardized, so that comparisons can be made between all my samples. If I fail to capture proper metadata, then this may lead to inconclusive conclusions down the line, or my data not really being relevant to the field. It can't really be compared to anything else. So this brings us to the main question that I hope we'll have answered by the end of this video. What are metadata? And that always trips me up. It's a plural thing, so. At its core, metadata are the contextual data that goes along the primary data that you are capturing out in the field. It's everything else that you are writing down or making note of, and it starts, when you go out there, the local environment, is what techniques you might be using as you collect your sample, and also everything that happens back at the lab as well. It's important that you capture this metadata not only for publication's sake, but also to ensure that your data is well supported and abides by FAIR data standards. As you may remember, FAIR data is findable, accessible, interoperable, and reusable. So by ensuring that you have as much quality metadata as possible, you are getting closer to having good, fair data. And with fair data, this makes it so that your sample data is findable by other researchers, and that they have enough context with it so that they can then use it for their own experiments, which, gets you hopefully another mention or at least a citation. And that's always good for people in sciences. So metadata, although I've been sort of talking about it as more of the environmental type, it can be found at every stage of the sample processing pipeline. We have sample metadata, preparation metadata, data processing, and feature metadata. Preparation, data processing, and feature are just as important as sample. However, this is more of what you find in lab, and hopefully you have a standardized protocol so you are able to more reliably capture the metadata. Today, I'm going to mainly focus on the environmental side and the sample side, because that's what has the largest, breadth, as well as, it is more likely that we're going to mess up capturing our sample metadata or capture it in a non-standard form. So, what should you include when capturing your sample metadata? Well, first and foremost, you're most likely going to want to go through the 4 Ws and I guess in this case, 3 W's and the how. So when was the sample collected? You know, this could be not only time or not only the date of it, but time of day, you know, where it was collected, the actual latitude, longitude, or just a description of the local environment, how it was collected, you know, give information on what tools and techniques were used, because that may play a role in changing how your sample data is actually analyzed down the line. You know, you're going to want to talk about what kind of sample it is, and then any treatments that might have been applied to it during the experiment. And finally, you're going to want environmental properties, which is a very loose umbrella of saying, you know, get as much as you can just so that you can sort of paint a picture of what, you know, things were like when you captured your sample data. And all this should come together to be enough so that someone not unrelated to the project can later visit it and have a good idea of the context behind your sample data. Now, in microbiome science, there is a large breadth of information that we have to cover, and you want to make sure that you're capturing as much relevant metadata as necessary to understand your project. And that isn't to say you should just go out and record everything, because then that makes it a lot more cumbersome on you and may lead to some slight mistakes. You want to make sure that you capture everything necessary to describe your project. So I know you guys have been working with a streams dataset, and within a streams dataset, you're probably going to want to capture metadata related to, you know, the stream itself or the water. So things such as the local climate, the pH of the water, any nutrients like carbon, nitrogen, maybe the depth, are extremely important to, you know, understanding the microbiome datasets within the stream dataset itself. However, things like maybe, you know, air quality might not be as relevant, and so that might be something you skip over unless it's extremely important to understanding, you know, the stream environment itself. So I've given you guys a sort of background on what metadata is, and now I'm going to transition into, you know, taking care of the metadata from the collection stage all the way through your project. Now, metadata capture and storage isn't over as soon as you collect your sample. It's something that is constantly evolving, and you need to be cognizant of the entire time you're working with your data to make sure that things aren't lost. Just like how you would create a data management plan, you should have a metadata management plan to make sure that you're not losing anything, and that when it comes time to submit your data set to a repository, you have everything already pre-filled out and you're able to describe every bit of sample data that there is. Again, this is to make sure that you don't, you know, lose anything and you understand the context, but also so that anybody later on who wants to access that data and might want to use it within their project can easily find it and make a quick decision whether it fits with their work or if it maybe is outside of their scope. Now, I'm going to give a quick example of a bit of metadata that might not be valid and complete. So this is something pulled from NCBI SRA, and within the file itself, there is a small metadata sheet that includes things such as sampling date, pH, geographic location, and so on. However, NCBI has its own metadata field, and these were not filled out properly or applicable. So if you try to look up this data set with any of the metadata fields on the right, you wouldn't be able to find it. Also, even though they submitted a metadata sheet, it's not completely valid. So if we take a closer look, we can see the sampling date, although it is present, does not align with the collection date. And that is due to a different format. The format that they submitted their sampling date does not follow the standards that NCBI has. Next on the list, we have pH, which is 100. And that is quite a basic solution and probably something that you would never be able to find. It is clearly here an invalid entry and should have been flagged before they submitted. Geographic location, they just left an NA, so that is a field that they just didn't fill out properly. Their host, they've listed as plant, and there are quite a bit of plants out there. A bit more information would have helped anybody trying to look this data set up. Elevation, 10,000 meters. This is probably the wrong units because that is really, really high, and I don't know of any experiments that are happening that high, unless you're, trying to go to outer space. And then for sequencing technology, that is just a missing field, completely blank. So why do we need to set these metadata standards? Well, by setting metadata standards, this makes it so that we can easily compare two data sets from different researchers to each other. Without a set standard, it may be hard to make these comparisons, or you may have to spend a lot of time standardizing everything, which takes away time from analysis or writing or whatever else, you have to do. I'm sure you're busy people, and the last thing you want to do is spend hours making sure that all the units match. So we have a little exercise here to sort of show you issues in standardization. In this exercise, we have two researchers working on very similar projects, maybe not the exact same field, but close enough where they would want to compare their data sets. However, they did not work together on creating a standard metadata set. And so when it comes time to compare, things get a bit tricky. I hope that off the top, you guys can notice quite a few differences, but we'll go through each one and at the end explain how we can sort of fix it. The first glaring issue that I found is the headers. We have a difference in how the samples are actually named. So one of them calls it ID number, the other sample num. Both probably mean the same thing, but if you were to directly align them, then just wouldn't mesh together. Again, we have the same thing with material and substance. And then we have temperature and temp. Both same things, except one is abbreviated. And so if you try to merge the data sets, might be considered two different fields. Next difference we see is the way that they call the actual material they're studying. So one researcher calls it soil, the other one calls it dirt. One calls it groundwater, and the other one calls it ground liquid. Now, I personally have never heard of ground liquid, but I'm sure that you guys can think of your own examples where a substance, which you think, is pretty standardized, someone's come up with a name, you just don't understand how they got there. Now, when looking at the sample depth, this is an issue that I find pretty relevant in my field because everyone has their own way of measuring the depth of their samples. Some people do it in meters, some people do it in centimeters, and in my field, which is agriculturally based in the US, everyone does it in inches. So if you're not paying attention to the actual units being used here, you might end up making a mistake when doing your analysis and, overinflate some numbers because, you left one in centimeters, another one in meters. Big no-no, and something that you need to catch early on, or it might make it throughout the analysis. Finally, we have an error in the way, or not an error, but missing information about the temperature itself. Now, the researcher on the right may instinctively know that 21.1 and 7 are clearly Celsius. There's no way of misinterpreting it, but someone may be working in Fahrenheit and just briefly glimpse over it and assume that maybe they're working in Arctic environments. That's why the temperatures are below freezing. And so they might throw an F on there, and now you lose a lot of context behind that sample. So it's very important to make sure that you are adding as many units as you can, unless specified otherwise. There are some submission portals that only accept things in Celsius and want you to just input the number, in which case you followed whatever repository rules there are. So in order to make sure that everything lines up, we can first use the headers on the left, transfer them over to the right, and then change the names and the units to align everything. And by doing so, these two scientists can now better compare their actual sample data, knowing that their metadata fields are aligned. Now, we need to talk about the consequences if you fail to standardize the metadata or just don't capture all the necessary metadata itself. And the most immediate effect that you might see from this is you miss out on critical context when you're doing your analysis. If you miss out on capturing temperature, you may miss out on a heat wave that came on 1 or 2 sample days that really pushed your data set to one side of the spectrum. Next consequence is that your data might not be accepted and published to certain repositories. Nowadays, when you submit your paper to a journal, you're going to need to submit your sample data itself. And with that, you're going to need to provide the metadata for that sample data so that anybody down the line who wants to use that understands where that data came from without having to read through your paper or contact you. Not having standardized metadata also limits the amount of comparisons that you might be able to do within a group of data sets. If you don't label your forest or streams data set as streams itself, then other people studying that streams data set wouldn't include it. Having non-standard metadata also prevents reproducibility, and most importantly, or not most importantly, but still pretty importantly, makes it difficult for other people to find and then compare it, which goes against fair data. So that is quite a bit about metadata in just a little bit of time. And I hope that you now have a better understanding of metadata and why it's so important for not only your science, but also for other scientists in the field. The main lesson that I hope you come away with is that metadata is just as important as your main sample data, and that without either one, you really can't make a good conclusion. If you have any questions about metadata or metadata standards, feel free to ask my fellow NMDC ambassadors. They're all experts on metadata. And if they can't get it, then they can provide you with my contact information, and I'd be happy to answer any questions you have.
Okay, so we did our break. We have heard from NMDC. I'm gonna go through the DRAM outputs that Jared and I talked about.
We're doing the questions first on.
Yeah, we're
Okay.
gonna do the question first. But I'm gonna go through the activity setup for DRAM. So we'll go through the outputs of DRAM just so you can see like what they actually look like. And then we'll be on to KBase and then lunch. So that's where we're headed.
Okay, the question online in the notes doc, are there templates available for preferred metadata formatting for sample collections? And the answer is yes, NMDC has templates. They leverage the Genome Standards Consortium Mix-S templates, which is the minimum information for any type of sequence. And I think we can provide some links in the document maybe to those resources for the online folks. And then the in-person group will have, are you doing water packages today? Okay, yeah, so the for the in-person people, you'll see the online submission portal. For the online people, we'll make sure that those links get added to the notes document so you can explore, and then you can do follow-up for any questions with the NMDC team later. Any other questions from online or in the room?
Clarification. So the templates, are they specific for the sample, like dealing with water, underground water, or soil or plant, or things, for example, something like that?
Yeah, they're done by environment. They're environmental packages. So it's typically if you're collecting a soil sample or a water sample, there's host associated. Yeah, so. Mhm.
Okay, so what we have done, and people online, we will make these narratives available probably sometime tomorrow, or when are we gonna make the?
Yeah, so for the online community, we will share a link to the organization. So we talked about this a little bit yesterday. There is an organization called GROW, Genome Resolved Open Watershed. The narrative that Kayla is going to talk through, right, that was this all Congo genome one. And then these are the in-person sort of group activities that we're doing. We will share a link to join this organization at the end of the workshop. And anyone with a KBase account can request to join, and we will admit you to the organization so you have access to all the data as well.
Okay, so right now, this is the KBase organization, and so you all we've been working in single samples, and now we're gonna transition. So what Ben has done for us is he has pulled in all the mags from all samples and put them into this all Congo genomes narrative. Oops, and that is right here. So this is the all Congo's genome narrative. And so this narrative is annotated with some questions that are going to be the scavenger hunt for today. So we're going to go through, we have taken these genomes, and by we, I mean Ben took these genomes, he dereplicated them, he ran check-m on them, he also ran GTDB and DRAM. So it's all been ran, and now the goal is how do we actually wade through this mad amount of data, right? We ran the jobs, they are all done for us, but we get all these spreadsheets and outputs, and so let's go through each of those outputs. And that is the goal of the DRAM activity today, but I wanted to walk through this narrative and specifically the DRAM outputs for in-person and online, so you can kind of see where you should be looking for answers to these questions. So there's about 15 questions, and for the folks online, these questions are not unique to this data set. You could work through your own data set using these questions just to get an idea of where things are. Okay, so step 1, run check-m. There's some questions about check-m. There's some questions about DREP. I'm going to focus on DRAM for now because there's a lot more different kinds of outputs. More questions. We'll scroll down to DRAM. So step 5, this is the last step. So run DRAM on your bin set. Again, these have been done, but these are the questions for DRAM specifically. And now we'll talk about some of these outputs. So if we go to this annotate and distill with DRAM that Ben has ran for us, you can see the product heat map that Jared was talking about. So you can see here, these are all your genome bins on the side that were generated. And so keep in mind, these are your dereplicated bin set. These are all the functions that he talked us through. And if you scroll over these, you can see what he was saying. So for example, if you're interested in this bin 12 FASTA, you can mouse over the glycolysis pathway, and you can see that this genome has 5 of the 9 steps for glycolysis. You can keep scrolling over and see, the different things here for thiosulfate oxidation, for example, and you can see what genes were called based on this. Okay, so that is the product. You can also, the other thing that's really important here, so remember how we told you that there are lots of levels to the data in DRAM. It's not just the annotations.tsv, the raw data annotations, it's also the product and the distillate. And so each of these files are output from DRAM. And I just want to stress, in any tool, not just specific to DRAM, anytime you run a new tool and you're not sure what the outputs are going to be, or you're not sure where information is, I would encourage you all to open all the files. Like, just starting with a new tool, open all the files, take note of where things are, because oftentimes things are hidden in different files because it made sense at the time to make it that way. And now you really need this information because that's just how tools get developed. So I really stress to all my students, just open all the files and see what's there. And then we'll go from there. But anyway, the files we're going to focus on today are the annotations.tsv. So that's the raw, and the metabolism summary, that is the distillate, and then the product HTML, which is that heat map above. You can download it from here. And then there is, sometimes I prefer to look at the product in a TSV format. So opening it in Excel so I can sort by like true or false on specific functions. So that is there as well if you want it. Okay, so Alicia opened these files for us. So Jared already talked you through the product heat map. Oh, cool. So this is, I just wanted to show you these files for the annotation. So this is the raw data, keep in mind. So what we're looking at here is, I'm gonna open these up. Open. Long headers. Okay, so this is, I'm gonna open these up. Okay, so we'll talk through each of these columns. So this is the annotations.tsv. So remember, this is all the data that you have is in this file. It's a big file. So it's every gene that was called and all the annotations for each database that you use for each gene. So that's a lot of genes, a lot of annotations. The first column is we'll start here. The FASTA is the bin that you input into DRAM. So this is the bin. Then each bin has several scaffolds or contigs. And so each thing in this column is a scaffold within this bin or contig. And then these are the gene calls on this scaffold. So this is gene 1 on that scaffold. This is gene 2 on that scaffold. You can see the underscores. If we scroll over, so this is just, the kind of logistical information of our genomes. Then this information here is our data from Prodigal. So the gene calling information. So this is the gene position. So again, this is gene 1. The start position of that gene on the scaffold or contig is at 3, and that gene ends then at position 1, 2, 2, 6. And then it also gives you strandedness. I'm going to skip over for rank for 1 second, but I'll come back to it. Then with the annotations file, it just compounds all the databases onto the end. So this is the KO-FAM scan. This is the MIROPS database, the peptidase database. This is the PFAM database. This is the CASI, Kzyme database. And then this is actually counting for multi-heme C-type cytochrome motif counts. If you're interested in things like iron reduction, you would go to that column. The last thing I wanted to touch on is rank. So each of the genes in these annotations are ranked A through E. And so if it's a reverse best hit on the annotation, so you blast your gene to a database and the return hit is the same, you get an A for KEG, and it goes down the line to PFAM and then so on. And then if you have no hit, for example, you would be an E. And the actual like A through E rank is spelled out in the DRAM paper. So that is the annotations.tsv file. And so again, this is the place where you would look if you have something that's not maybe isn't already in the product or something that's not in the metabolism summary. This is also the place you would go if you, for example, wanted to build a tree of like a PMOA, a methane monooxygenase tree. I want to like make a tree and see, you know, how they're related to each other. You would go here, sort this file, and then pull those genes out of the genes file on KBase 2. Okay, so that's the annotations. And then I also wanted to quickly show you the metabolism summary. Okay, so this again, so we had the raw, now we're in the distillate. So this is one level up from the raw. This has been summarized a little bit, but not as summarized as that product heat map. So this is, you can see along the bottom here, so that figure that Jared showed where it had, you know, all the stuff in each. So it was like a miscellaneous tab. So things like flagella are in the miscellaneous tab. Carbon utilization, here's all the CASIs. Transporters, all the transporters are in the transporters tab. Energy metabolism, organic nitrogen, and so on. And so all of these tabs have the same format. And so genes are along the side. And then these are genomes. And as I think somebody asked a question, well, how do you see the copy number of specific types of genes? That would be found in this file. So you can see this one has, you know, 4 copies of this carbon monoxide dehydrogenase, while this one has 3. So all the genomes are along as columns, and then the gene types are here. One thing that's handy about this metabolism summary is if you're going to look at, so I really care about energy metabolism, but I don't really care about all the energy metabolisms. I really am interested in something like nitrogen. You can then, we've set up these headers here and modules, so you can filter out the things that you care about. So there's, they're further broken down beyond, you know, miscellaneous, carbon, nitrogen, and these things, energy. You can then say, okay, well, I care about energy, but I also really just want to know about nitrogen metabolism. Then you can sort this thing, and now you have all the nitrogen, I'm on the energy tab, sorted by nitrogen, and I can see all the genomes with the capacity for nitrogen metabolisms easily, and it's counted and summarized. And then just to stress one more time, the next level is this product summary that Jared was going through. And so hopefully that sets us up for our questions. So the activity for DRAM today is you all are basically going to go into this Congo genomes narrative and work through the questions that are at each step. So there's questions on CheckM, there's questions on DREP, and there's questions on DRAM. And then we'll be circulating to help you find those answers so that you know when you're working on your own data sets where you can go through and find the answers that you're looking for easily. Okay, so I'm gonna do another check. I really like crossing things off lists. So we're done with the activity setup, and I'm gonna hand it over to Ellen.
Hello, everybody. My name is Ellen Dow. I'm part of the KBase user engagement team. I also lead our educators program, which I'll give a little bit of shout out later. Today, I want to talk about how you can really utilize KBase and make some really great narratives, some shareable workflows to improve your publishing or really help make your work viewable and usable by others. So you saw this figure yesterday in day 1 when Ben was presenting about just what KBase is, where it comes in. Again, within the research data lifecycle, you've gone out, you've collected and processed all your samples. You have all that environmental sample metadata attached to those samples. So you have all that great information about the data that you can then bring into KBase, start analyzing, doing some comparison, really building your own analysis workflows, working with your collaborators, accessing different tools, and then you can start publishing your results as part of your curating and publishing that data. And this sort of comes in KBase with right now we've been working in the narrative but we also have what are called static or fair narratives. And these are viewable outside of KBase itself. So they're HTML snapshots that allow you to share your work with those outside without logging into KBase itself. And so I want to just kind of go back to some of the points around why you might, not share your data, or what might prevent researchers or yourself from preparing and sharing your data and metadata. And so we have kind of three big components here. The first being knowledge barriers. This is very much of just the, why would I really want to share my metadata and data? I have a really, really large data set. So how do I actually get it out there to others? Where do I put it? There's also concerns over reuse. Am I going to get credit for putting my data out there, or is someone just going to kind of scoop in, take it, and not say, oh, yes, this person collected it? So that's one thing. It might have sensitive content. You might also be kind of afraid that somebody is going to take your data out of context, without that metadata information connected to it. And then also, some of the other disincentives of, scooping. It can be time intensive. You might not see the worth in sharing your data if you aren't getting credit for putting it out there for others to use and compare to. So we are actually working on that in KBase. We have a wonderful credit engine team who've been really working towards thinking about how we can ensure that the data that you are putting on KBase and if you decide to make it publicly available to others once you're ready to publish, or once you have a clean data set, that you can also receive credit for putting it out there. And so in KBase itself, we do track data provenance. So that means that if your data is within the system, we can see how it's been transformed, what tools have been used to analyze the data. So both like within your workflows and by others. And so KBase itself, we are really working on leveraging external resources to make sure that your research products, your data are fair, so that findable, accessible, interoperable, reusable, as well as trackable. And so we're really thinking about data, what the data is itself, who created this data, whether it's you or if you shared it with your collaborator, who is using it there, and then how it was generated. And so in this little model that we have, it sort of represents two different researchers who are using KBase. So we have researcher orange, researcher 1 in orange. And so they have added their data into KBase, and they are creating their own narrative, their own workflow, analyzing that data, and also pulling in some public data. You can pull in data from the Joint Genome Institute. We also work with other Department of Energy partners, such as ESS Dive, NMDC, of course. There's the NCBI database if you have SRA reads that you want to use to do some data comparison. You use the different apps and tools that are on KBase, really build out that workflow. And then what you can do is you can produce a static narrative, so a nice HTML snapshot. So that is a version of your workflow at that moment in time that you're able to share out. It's also really nice because when you spin this up, when you've made that narrative public and made a static narrative, you also get all the tool versioning information. So all those citations are all in one place, as well as all of the different data objects that are within that particular narrative. So you have all the information on the data products that are being shared there, as well as the tools that have been used. So you have created this narrative that's viewable outside of KBase. Then we have researcher 2 or Teal that saw that narrative and said, hey, this is a really cool workflow. I like this. I want to use this workflow and maybe use it to compare some of my data to. So they can make a copy of that narrative. They're able to take the data, take the workflow, exactly how you did it, see all the parameters, how you analyze that data. They can import it and make their own copy, import their own data, make their own copy, do some comparisons there. And then they can create their own static narratives, that fair narrative, which automatically will cite your orange researcher, their narrative, so their work. So this way, we're ensuring that if you have shared your workflow, how you're doing your analysis, your data, it automatically is cited by someone else who uses it. So it really takes away some of those barriers that might be, well, if I put my data out there, how am I going to know who has used it, how it's been used? And we can help tell you how it's been used, and exactly what tools have been used, when it was, when that analysis was done, all this information. So one of the things we do is we can also track these research projects by persistent identifiers. So you probably are familiar with the DOI, digital object identifier. You always see those DOI links when you look at publications. There are a few other persistent identifiers that we can also talk about. So we have our person in red, and we have a publication. And so we know that is where our DOI comes in, but we also have all these other items that are attached to one DOI. And this includes the different components where we have, our sample, our protocol, data set, the software, the publication itself. All of these are linked to one another, including all our funding, how we're accessing and using that data with our data management plan, which NMDC had spoken about earlier today. And then we have so many other persistent identifiers that are also linked here. So we have that research activity identifier, IGSN, or the International Geo General Sample Number. So that sample itself, all the metadata and information, making sure that that is connected with your data set, with your publication, the software that you're using to analyze it, your own persistent identifier, oftentimes, especially if you have KBase. You can link your ORCID account so that you have that identifier that is attached to the publication, all this information. So we're able to link and keep these things all together with KBase. And so one of these examples we can look at is GROW. Kayla's been talking about her project and a lot of different work. So in this example, we have Kayla, who's at Colorado State University. We have her ORCID. You can see the funding resource from the Department of Energy, and exactly the particular project that this was funding came through with the JGI CSP proposal alongside that data management plan. And then within the actual project itself and that workflow, we have the research products, including, the sample. You can see the IGSN that's there, the protocol ID, the software ID, the bioarchive, because right now the publication is in revision. And then we also have the link directly to the workflow or that fair narrative itself. So all of these components are interoperable and you can track them together. We're trying to package it up like a nice little package. So instead of thinking of them as all separate pieces, you can look at them as one. And so by having your data within KBase and doing this analysis, you can also think about how your data itself can be tracked to publications. And this is where we get back to those persistent identifiers. And with KBase, having a DOI attached to your narrative that you can then cite within your publication itself. So you have those two digital object identifiers that are connected with one another. So you can also tell where your data has been reused, how it's been reused, so people can really find that data and connect to it through your publication versus having those as two separate entities. And in KBase itself, we can look at how much that data has been viewed, as well like what's happened to it. So for this example, particular, we have 136 people have looked at that static narrative, and then within KBase, 6 different users have created 17 narratives from that one narrative. So they made copies. And then also we have the generation of 3 new data products. So more analysis that has been done, further analysis downstream. So this is a great example of data actually being reused within as our last fair principle. And so how we're doing this within KBase is you have your KBase narrative. You can then request a DOI, which we do through OSTI. The narrative will get that DOI, and we do it quite nicely. We keep that particular narrative numbers, that workspace ID. So you see this 109,073. We keep that the same as within the data DOI when we request that. So there's a nice contiguity to that. And then you cite that particular DOI, you cite your narrative within the publication. This is also linked to your own individual ORCID, so it's linked to the person itself. And then again, we have that fair narrative and all those different data objects that are spun up within KBase. So tracking what's happening with that data and where it's going. For another example of this, from a different perspective, we also have instructors who are publishing with their students. One of the data publications is the American Society for Microbiology resource announcements. So if you have an isolate genome, or what we've been talking about here, metagenome assembled genome that you are ready to put together and publish, whether you yourself are doing this or with your students. We have an example from Siena College. So these are undergraduate students, and they all went through, so a class of, I think, 15, if we count Anna, our instructor, as the 16th author. They went through and isolated 6 different species of bacteria, and then they have 6 different narratives, which are all attached to our parent narrative. So we have a single DOI that parent narrative has, and then we have each of the isolates that are associated with that. And again, we have the same workspace ID that's connected with that DOI when you have the digital object identifier that we attach to that narrative. So this is one example with ASM-MRA, and we actually have a really great partnership with them where KBase has been set up as a repository. And now we also, for those of you who do teach and are interested in using KBase with your students, we have a KBase educator, I guess it should be educator and students because these students are also authors. We have a special collection with MRA, and I'll show you that page in a little bit. Because I am talking about MRA and how we're using it and thinking about how we can make it easiest for users of KBase to come in and go through, we do have a template. I will go through the metagenome assembled genome one, but we do have an isolate template as well. Really what's within this template are a lot of tips, help, resources. It takes you exactly through that checklist. So if you go to MRA to do the publication itself, that checklist is within the narrative. So you have an introduction, the background, experimental methods, the import, quality control, assembly, annotation. All of these should sound very familiar to the workflow that we talked about yesterday. Taxonomic identification, and then you can also go into some metabolic modeling, flux balance analysis, and then references, which again, when you spin up that static narrative, you have all the tools and their versions cited, ready to go. And then we also have some final steps for submission as an MRA publication, including some of the non-KBase requirements, such as how to get your data into NCBI, so if you're submitting to SRA and such. And what this narrative looks like is here we have a static version. So this is that HTML snapshot. So it looks a little bit different from the narrative itself when you're logged into KBase. Here is all the narrative. These are all markdown cells, which are spun up here. So within this workflow, you have, the background information, and here we have the particular background information about this template and why the template exists, as well as some instructions on how to use it. And then we have our nice table of contents. And then it goes into really what information should be included within your microbiology resource announcement. And this goes from, the title, abstract, introduction, and then goes through the checklist where we have information about the sample itself. If it was isolated, any genome sequencing information, and then into the different steps and how it was analyzed. And then those final steps with getting exactly how to get a DOI, how to use the dataset DOI, linking that to your publication DOI, and informing you that you also need to put your data in NCBI. So this is that snapshot version. We can also take a look at the narrative version itself. So this is what you go to, and you would click on this copy button. And let me also just make this a little bigger for everybody. There, that's nicer. Is that good? Okay. So here is that version. Up here is that copy button. You can see it's in view only mode. So I can make a copy, and then I'll have edit permissions. So I can use this template, add my data to it. So add my raw data, start going through, adding my quality control tool that I want to use, in our case, FastQC. Then going through, adding the assembly application that I want to use. So whichever tool you want to use there, annotation tool, and so on and so forth with how you decide to analyze your data. And again, we have these nice markdown cells that have all this information. We also have some tips, how to use Markdown, how to use the language, do some different formatting. You can see here, we have some nice headers. We have hyperlinks. So we have tools and tricks on how to do that within KBase, both within a narrative that you can make a copy and you can see all the background, exactly what the Markdown language looks like, as well as within our documentation site, docs.kbase.us. And then again, just to repeat myself a little bit, show you again what it looks like in the narrative. As you scroll down, you can see you have the author checklist, all the background experimental methods information. So how did you get that either water sample, soil sample, how did you collect all that microbial information? And then exactly what metadata might be attached to that. And then you can go through with the quality control, assembly, binning, annotation. We do have some nice hints. Zach has been really great and put in a little bit of comparison with what different applications you can use for quality control, as well as binning, since we are talking about here metagenome assembled genomes. And then a little bit more on, taxonomic identification. And, Zach put in a nice little note of using the GTDBTK classify app. And then also you can put in a phylogenetic tree if you want to. Then it just kind of goes through references, which you will automatically spin up when you make a static narrative. And that, I can't remember if we showed how to do a static narrative, but when you're doing that, what you do is you go down to share, and you can go down to, this one's public, so I don't have to make it public, but you can go down to managing static narratives. And then there are 4 versions of this. This has existed for a while and it has been updated. So you can update and make this static narrative, but it's just through that share button. Okay, so the last thing I'd like to just share out with all you is the MRA collection that we have for educators. There we go. So we have this nice collection of KBase being used in the classroom or lab, whether it's a wet lab or purely a dry lab, if it's just bioinformatics, or in silico lab. We have a little bit of information about this, and then you can see we have all the different featured articles from these different instructors, and you can see all their students who are authors. So we're really trying to make sure that as students are actually doing this work, they're going through, they're learning the process, but they're also adding and contributing their data to, you know, to this environment, really demonstrating those fair data principles and sharing their data out. So with that, that's all I have for you today. Are there questions?
I was just wondering, like, if you could talk a little bit about if you have experiences using these tools with students and what are some like common challenges that you've faced?
Yes. And not just me, but I have input from a lot of instructors. We will send out some information about the KBase Educators Program, so I can send all of the tips and tricks, but we do have an educator handbook that walks through a lot of what instructors have seen. I will say, I think one of the biggest, or like one of the first ideas that you want to really instill with your students and just KBase itself is that KBase is a platform. Like it is a place that you are using analysis tools on data. A lot of students like to just say, we use KBase. No, you did like the work in KBase, but you are using all of these different analysis tools for assembly and annotation. So that I know is one of the comments I've had a lot with instructors, is just really instilling that information, as well as just, you know, kind of the concepts of, you know, what is a genome, you know, how it's all the sequences, the annotation, that like information about that organism. That's my one of my big top level answers. And then I have resources. And I guess for anyone, if you are interested in joining the KBase Educators Program, please send an email to myself or engage@kbase.us. Let me know that you want to join the educators organization. So what we have is we have a beautiful landing page, as well as our handbook, the KBase Educators org, which is within KBase itself. And I can give you the information on exactly how to join that. So you can access a lot of different teaching workflows, because what we've done is we've taken these big workflows and we've broken them down into chunks. Oh yeah, please. I can also look it up. Oh, yes. Yeah, I'm getting a question. Yes, Arjun.
Yes, so you mentioned that landing page. Is that on the KBase website, or is it a different website from your university?
Yes, it is on the KBase website. If you go to kbase.us and then go to, thanks Alicia, engage, it is linked there, or Alicia just put it in the chat.
Thank you very much. Yeah.
And we do have educators all over the world. And I'm pulling up my map of where everybody is. Okay. Yeah. And let me put this into slideshow view. There we go. I don't have that many educators in Africa, so if you want to join, please do. But we do have instructors all over the globe who have been using KBase with their students, or at least that I know who have like joined the org and I have contacted and chatted with. But right now there's actually, I think we're at 199. So if you request to join my org, maybe you'll be number 200. I guess we'll have to like, I don't know, balloons will have to like fly down something. Yeah, but right now there's about 100, almost 170 institutions. We have all the way from high school instructors, instructors at community colleges, primarily undergraduate institutions, as well as quite a few minority serving institutions and emerging research institutions. And these, I know it's not as applicable for our international audience, but within the US, especially with Department of Energy funding or National Science Foundation funding, there are a lot of programs that are there to support instructors at these different types of institutions that don't have all that funding that an R1 or a research institution might have. Yes.
Question. Regarding just the platform and the training as well, is it offered only in English or?
So, no, that is a great question. So right now it's primarily in English, so share documentation. We have been working with building out and taking some of our teaching workflows and translating them right now into Spanish. If you do have a student who's really interested in bioinformatics and thinking about how they can make it accessible to others in their native language or one of the languages that they speak and would like to make that a resource, I am happy to work with them and kind of set up a working group where we can translate more of these resources into another language.
So you like African map, most of us on the same time, we speak French.
Yeah.
So great to have this.
In French, yeah.
Yeah, and we will, so for students especially, we can offer an honorarium.
Yes. So
It's not free service.
oh yeah, so the interns who have been helping me translate into Spanish, they have received honorariums and a stipend for the work that they put in.
I think internationally it would be hard to set up intern appointments.
But an honorarium.
Do a one-time honorarium.
Yeah. So for those online, just in case you didn't hear, if you do have a student who's interested in translating some workflows or any resources, we can set up an honorarium for those students. To give them credit for, I mean, you put in all the work, you really, you really deserve something. But it's nice. Like they also, in these narratives, these interns do have a credit on the narrative itself. So they are listed as one of the authors. I think we can kind of, oh yes.
Not a question to say, but more comment. So, I mean, I'm a community ecologist, and I'm glad that at the DRAM outputs, you can get different files where you can perhaps do like some alpha diversity, like Shannon and richness. But I think while it's great to expose students to such platform, but they also need to learn to do some, descriptive stats and all of that kind of thing. So I think, to give them that handwritten stress that they share and take. I think it's really great that we can have such outputs, and while they're looking at the functional diversity, whether nitrogen and so on, they can do some, because it's good to know that it's there, but then also obsessed with the significant that kind of thing.
Yeah. I mean, I think there's so many different applications and ways that you could really take the resources and make them your own. And that's our goal. is to, you know, have something that's much better than nothing to start with. And then really be able to go and dig deep once your students are at that level. And I'm happy to also work with more folks on thinking about how we can adapt and, you know, build in more layers and levels of just like ready to go packages for instructors. I think now we have Q&A for everybody. So, yes, Arjun.
Yes. So it's more a general question because I noticed that these presentations were also recorded. Will those recordings be available?
Yes, we are going to make the recordings available after the workshop.
And will that be through YouTube or would it be from
Yes.
your local website?
Yes, we're going to put them on YouTube, and we will also do a follow-up. So we'll send an email to everybody so you'll have all the links.
Oh, excellent. And yeah, my question about participants in the Netherlands, because I'm very interested in this teaching program, but I just want to see if somebody else already uses it in the Netherlands. So I will be very happy if you can send the names.
If you, I was like, if you join our educators org, you might be able to find them in there.
Oh, okay. Is that so? If I join, then I will see the overview of all the participants.
Yes. Yes.
Okay, very good.
All right, we're good. Okay. All right, thank you everyone online for joining, and we hope to see you during the week at ISME if you're here.